Let me start with the origin, why we started looking at the gut in type 2 diabetes. And it's about the incretin effect. So this is from an early study published in 1986, and it's what everyone knew at that time. If you measure glucose, insulin, C-peptide in healthy subjects and compare that to type 2 diabetic patients, the glucose is going to be higher in type 2 diabetic patients, and the rise in insulin following oral glucose does not follow this steep course that is normal in healthy subjects, and that can also be seen when you look at C-peptide. If you want to know what the contribution of the gut is, if you want to measure the incretin effect, you do a second experiment, infusing glucose, and I hope you follow the rationale of this experiment that the glycemic stimulus to the pancreas, to the beta cell, should be identical under both conditions. So whatever glucose does to stimulate insulin secretion is the same. And if you then, surprise, find a big difference in insulin and also in C-peptide concentrations, much more with oral glucose than with intravenous glucose, then additional factors must play a role to explain this incretin effect. And I want to point at this point in time to the quantitative difference. So it's really that 2/3 of the insulin response to oral glucose is not mediated by the rise in glucose, but by something else. And this something else are incretin hormones, and in humans we know two of them. GIP, originally named gastric inhibitory polypeptide because it was discovered as something inhibiting gastric acid secretion, which probably is not an important physiological function, but you can now reinterpret GIP as glucose-dependent insuliontropic peptide, so you keep the abbreviation GIP. And the other one is GLP-1, glucagon-like peptide, which does not behave like glucagon at all, but it is similar in molecular terms, hence the name. And what you see is if you give IV glucose, the levels don't change in both cases. You have a low basal concentration, you have a steep increase to a peak after about an hour, and the response lasts up to 4 hours. I will point to the fact that with this scale, the peak level of GLP-1 is much lower than that for GIP, and this, among other reasons, is why I believe that GIP is physiologically the much more important incretin. GLP-1 probably plays a very minor role in physiology, but it is much more interesting in terms of therapeutic potential. So I may quote my former boss, Werner Kreuzfeld, who in his Claude Bernard lecture, 1978, had this figure. It shows the gut, the mucosa, and these cells, single cells in the mucosa that produce hormones. And if you look in the duodenum and upper jejunum, then it's K cells producing GIP. And if you look in the lower gut, then it's mainly L cells producing glucagon-like peptide number 1. And this is 1 out of 3 ways how the gut talks to the endocrine pancreas: endocrine transmission versus substrate and nerves. The nerves probably don't play a big role in this respect, and we know that because type 1 diabetic patients who receive a pancreas transplant, which is denervated, have a perfectly normal incretin effect. So what happens in type 2 diabetes? We can do the same experiment, so technically you can copy the glucose curve as well. You need a little bit more of glucose, which as a sum comes close to the 50 grams that you have given orally. In the healthy subjects it was much less glucose than the 50 grams that were administered orally. And then you can again measure insulin and C-peptide. And now you notice that this difference is much smaller than the difference that you have obtained in experiments with healthy humans. So it tells us that there is something wrong with the ability of incretins to stimulate insulin secretion. when you look at C-peptide, and in these years we believed that C-peptide was the better measure for insulin secretion because insulin itself may have a variable hepatic extraction or elimination in general. So there was not a single time point with a significant difference. Our conclusion was the incretin effect is reduced in patients with type 2 diabetes, and the question is why. So there are two general possibilities. Possibility one, there is something wrong with the incretin hormones as they are secreted from the gut in response to nutrient stimuli. So let's look at GIP first. And this is the largest study that I've found in the literature. So it's like 65 patients, and they both received oral glucose and mixed meals. And in both cases, the GIP response was, if anything, a little bit larger in the patients with type 2 diabetes than in healthy controls. And that tells us that a lack of GIP cannot be a good explanation for the lack in the incretin effect. So we did a recent study, and this is also a large group, so 29 healthy subjects and 32 type 2 diabetic patients. This study has the strength that each subject was examined 4 times under different conditions, and the different conditions were that one day they received placebo, gray symbols, one day they had received 10 days of vildagliptin, DPP-4 inhibitor treatment, and then the same experiments were repeated with an inhibitor at the receptor of the GLP-1 receptor. And I just want to ask you to compare the pattern in the healthy subjects with the type 2 diabetic patients. It is basically the same pattern, it's the same scale, so it's the same quantities, and I think this underscores that there is nothing different in terms of GIP secretion between healthy subjects and type 2 diabetic patients. How about GLP-1? There has been one large cross-sectional study by our colleagues in Copenhagen, with whom we cooperate very intensely. So May-Britt Toft-Nielsen did this study, and it's again like 65 patients with type 2 diabetes, a few with impaired glucose tolerance, and these are the healthy subjects, and these are the type 2 diabetic patients. And what she finds is low basal, increase after a mixed meal, and from the second hour on, some differences with less GLP-1 being measured in the patients with type 2 diabetes, and some intermediate values for those with impaired glucose tolerance. And I very often have heard a story translating this cross-sectional study into a longitudinal study. So it says, if you are healthy, then you have a good GLP-1 response. If you have early type 2 diabetes, then there is a little bit of a lack of GLP-1. And if you have long-standing type 2 diabetes, there is no GLP-1 secretion at all. And these are stories that can be modulated with a commercial touch, because if something is lacking, then it's of course very easy to explain why you should replace something that is lacking. So this is in the case of GLP-1 receptor agonists. And of course you shed doubts on the ability of medications that modulate the incretin system, like DPP-4 inhibitors, that they can have a clinical effectiveness later on in the disease. And for those reasons this is important, and so there are more studies than this. So my good friend and colleague Juris Meyer in Bochum did a study, and very carefully in like 30 healthy subjects and type 2 diabetics gave oral glucose and a test meal, and this is GIP and this is GLP-1, and he couldn't confirm May-Britt Toft-Nielsen's story, so there was no difference in the pattern of secretion. GLP-1 and GIP secretion were normal in the type 2 diabetic patients and the impaired glucose tolerance subjects. In our recent study, we again looked at GLP-1, and This is healthy subjects, this is type 2 diabetic patients, this is the total level of GLP-1 with a non-specific assay. This is intact, so this is not attacked by DPP-4, but the majority is in this fraction, and if we want to measure secretion, we look at the total GLP-1. What you can see is that total GLP-1 is reduced by DPP-4 inhibitor treatment, a feedback inhibition at the level of the L cell. And if you interfere with this feedback inhibition by infusing a large dose of a GLP-1 receptor agonist, then this augments the responses. But again, both in qualitative terms, the pattern, and in quantitative terms, what you see in patients with type 2 diabetes is very much alike what you see in the subjects who have a normal oral glucose tolerance. Again, no difference. We did a meta-analysis that was published in Diabetology last year, and we looked for studies that have examined healthy individuals and patients with type 2 diabetes. And because of assay variability, we took the healthy controls as 100% and then expressed the results in type 2 diabetic patients as a percentage of that. And what you can see, studies with oral glucose as a stimulus or mixed meal as a stimulus, you find everything. Hypersecretion, hyposecretion. This was the cross-sectional study that I talked about, but it is not at all representative if you look at the other studies published in this area. And if you do a formal meta-analysis to look for a difference, then the confidence interval is much wider than the small deviations from zero that you see for the mean values. So the conclusion is that neither in the case of GIP nor in the case of GLP-1 there is a lack of incretin hormone secretion in patients with type 2 diabetes. So this sort of answers the question whether the origin of type 2 diabetes is in the gut. That is probably not the case. But how the organism handles these incretins, how it responds to them. And remember, the main task of an incretin hormone is to release insulin, so to be a stimulus to the beta cell. So whenever glucose levels are above baseline, it should augment the insulin secretory response when there are elevated levels of incretin hormones available. So let's turn to GLP-1 first. And the question that I'm asking right now is, how does the beta cell respond to GLP-1 when you compare healthy subjects who respond normally to type 2 diabetic patients who may have an impaired response? And the answer is yes, there is an impaired response. And I have to explain this figure because I'm sure you will not understand such a complicated unit on first glance. So what Lise Kjemps did in this study, she performed different experiments, basically infusing different levels of GLP-1. So this is slightly above physiological peak levels, what you achieve with 0.5 picomoles per kilo and minutes. And then she infused glucose so that the concentration went up like a ramp. And then she related insulin secretion to glucose levels in these experiments. So what you get is kind of a slope. How much GLP-1 makes the slope steeper, relating insulin secretion to glucose concentrations. And at any GLP-1 dose, and the pharmacokinetics of GLP-1 are not different in type 2 diabetic patients than in healthy subjects, so this also applies to the GLP-1 levels. This slope was steeper in healthy subjects by approximately a factor of 4. So you can conclude that in patients with type 2 diabetes, you only have like 25% of the responsiveness to GLP-1, to exogenously applied GLP-1. So there is some resistance of the beta cell to respond to GLP-1 in patients with type 2 diabetes. But I don't consider this is the important message, and I'll come back to that later. How is this in response to GIP? The first study that addressed this question used porcine sequence GIP, which is a little bit different. So it's very difficult with porcine GIP to find the infusion rate that matches the endogenously secreted GIP, because the antibodies used in the assays react with different affinities to the porcine material and the human material. But the conclusion was right. Even in 1988, Tuor-Karup concluded that the response of the beta cell to GIP is massively reduced. And I just show you a more recent experiment where we not only considered GLP-1 and GIP separately, but we also asked the question, what happens when you administer both at the same time? So it's a study in type 2 diabetic patients, and you infuse GLP-1. Whenever you do it, you get a steady state of elevated levels, around 120 picomoles per liter. So the peak after oral glucose would be like 25 to 40 picomoles, so this is pharmacological. And you can also see that when you co-infuse the red symbols, that doesn't change the level of GLP-1. If you look at the active levels, you see that they are much lower, so like 15 to 20 picomoles per liter. So this is only 10% of the GLP-1 that is circulating, that is in the unattacked form, which is biologically active. And with GIP, we used a very high dose. The level is like 500 picomoles per liter. And in the case of GIP, the intact level is 200 out of these 500. So it's 40%, which tells you that DPP-4 attacks GLP-1 much more avidly than GIP. So GIP is more resistant to DPP-4 activity, and you can read that from these figures here. So what happens to glucose, insulin, C-peptide, and insulin secretion calculated by deconvolution? The blue line is GIP, and you can hardly reduce glucose significantly versus placebo with even a very high dose of GIP. GLP-1, green lines, normalizes glucose as in previous experiments. So after 6 hours, even after 4 to 5 hours, you have glucose levels of around 5 millimoles in the patients with type 2 diabetes. And the red line, you hardly see it because it's superimposable to the green line. GIP added to GLP-1 does neither make it more effective nor accelerates the fall in glucose. And this is mirrored, let me talk about insulin secretion here, by the insulin secretion data. So hardly any stimulation with GIP, and certainly no augmentation of what you see with GLP-1 alone with the combination. So basically what this tells us in terms of insulin secretion, even a very large dose of GIP is ineffective in patients with type 2 diabetes, and this is a uniform finding of, I would now say, around 6, 7, 8 studies that you can find in the literature. It is interesting to look at glucagon in these experiments because GLP-1 is known to suppress glucagon, and that was reproduced here with the green lines. But when you co-infuse GIP, which itself gives the highest glucagon levels and probably is able to stimulate glucagon under certain conditions, then this suppression of glucagon is counteracted by the concomitant infusion of GIP. So this tells us it does not only help to control glucose, but it may rather be untoward in if you want to lower glucose, because the suppression of glucagon contributes to the fall in glucose. And the free fatty acids are only suppressed in those experiments where you infuse GLP-1 and is not affected by GIP. So we can conclude that there is a little bit of resistance to GLP-1, but beta cells in type 2 diabetes still respond to GLP-1, and that there is a marked resistance. I would almost call it an unresponsiveness of the beta cells to GIP. And the question arises is, when in the course of the disease does this occur? Is this a prerequisite for becoming type 2 diabetic? Does this mean it's a genetic trait, for example, you don't respond to GIP and then you become diabetic? Or is it secondary to the development of diabetes, whatever diabetes means? And to address this question, our colleague Tina Filsbøl in Copenhagen, who works with Jens Holst, has studied this phenomenon in a variety of different etiologies of diabetes. Diabetes due to chronic pancreatitis, the rare lean type 2 diabetic patients, those patients characterized as LADA, so with autoimmune phenomena like type 1 diabetes, but on first sight they look like a type 2 diabetic patient and they still have some insulin secretion by themselves, MODY diabetes, and early type 1 diabetes where there still is some beta cells left. And what they describe in these experiments, you see a hyperglycemic clamp with the infusion of either GLP-1 or GIP or just the clamp. And what they describe is always the same pattern. So in these different forms of diabetes, beta cells respond to GLP-1. They do not respond to GIP, the triangles, and the lowest response is always seen with the clamp alone. And their conclusion, of course, is that if this phenomenon can be described in every case where diabetes is diagnosed, no matter what the etiology is, then this must be something that is secondary to the development of diabetes. And the reason may be hyperglycemia, it may be some other metabolic abnormality associated with diabetes. Along the same line, Philipp Knoop from the same group measured the incretin effect, much the way that I introduced to you earlier in this talk. And he looked at patients with chronic pancreatitis. As you know, if it's severe, they have diabetes. If it's not that severe, they have normal glucose tolerance, depending on the destruction of the organ and the affection of the beta cells. And what he sees is when patients with chronic pancreatitis have diabetes, then the difference is small, the incretin effect is reduced. And when they have normal glucose tolerance, then they have an incretin, much like comparing type 2 diabetic patients with normal glucose tolerant subjects who don't have exocrine pancreatic disease. So this would be the normal incretin effect, and this is reproducing our old finding of the reduced incretin effect. So what does an animal experiment tell us? This is in Fafa Zucker rats, and you can get at the pancreas and look at islet GIP receptor messenger RNA, and in heterozygous animals, wild type, you have a high level of expression of the receptor, and in those with diabetes, you have a low expression of the receptor. There is another experiment, a different model, 90% partial pancreatectomy in rats, and this causes diabetes. And when you look at what happens after pancreatectomy, then you see that the GIP receptor expression here in the normal animals is very much reduced. If you do the same pancreatectomy and lower glucose by fluorescein, so it's an SGLT2 inhibitor that you can use in animals but not in humans due to side effects, but in animals you can do it. So with a reduced glucose level, there is at least a partial normalization compared to what you see in healthy subjects. And you also see that the GIP receptor and insulin are co-localized, so we are talking about GIP receptors on beta cells in these experiments. Our friends in Copenhagen, Patricia Heuberg in this case, she addressed this in humans. And first of all, she reproduced the experiment that I've shown you previously, that now with somewhat lower infusion rates of GLP-1 and GIP. In healthy subjects, both incretins elicit a very marked augmentation of insulin secretion. This is when you just clamp and look what happens with hyperglycemia. So they were successful in finding doses that have an equivalent effect on insulin secretion. And if you do the same experiment in diabetic patients, type 2 diabetes, and just notice that this scale goes up to 400 and this goes up to 4,000. So it's really showing you that nothing happens at the beta cell, and both GIP and GLP-1 are hardly able to stimulate insulin secretion in patients with type 2 diabetes at these doses. She then used intensified insulin regimens for 4 weeks and brought the glucose down to these profiles, and I think this is as good as it gets. So probably in a 4-week experiment you cannot achieve a better glycemic control than that. And then she repeated the experiments, and this was what you have already seen before glucose normalization, and this is after glucose normalization. And then there is a slight recovery of the beta cell to respond to both GIP and GLP-1, but remember, the scale in healthy subjects went up to 4,000, and we are still talking about some effect, which is not negligible, but it is certainly not a normalization. And then you can ask the question, if hyperglycemia really is the culprit, how long does it take to take away the responsiveness to GIP and GLP-1, and how much would it take to regain the ability to respond to incretin hormones in those beta cells? So the real question now is this disturbed insulinotropic activity of GIP cause or consequence of type 2 diabetes? And to that purpose, Joris Meyer, a former doctoral student who now is a very successful independent researcher in Bochum in Germany, and myself, we analyzed studies that we had done over the years, and we looked at healthy subjects, first-degree relatives of type 2 diabetic patients in whom some predisposition might be visible, then women who prior had gestational diabetes and patients with type 2 diabetes, so they span a wide range of fasting glucose concentrations, and in all of them GIP was administered, and we again express the insulin response as percentage of what we find in healthy subjects. And then you very clearly see that below glucose concentration of approximately 126, you have a responsiveness to GIP, and above that fasting glucose, it's lost. That happens to be the diagnostic criterion for type 2 diabetes. So our conclusion from this is, whenever you reach the glucose intolerance that we call diabetes, then the responsiveness to GIP is lost, and it may be reduced in the impaired fasting glucose range. You can also look at the incretin effect in those studies that have looked at that, and you see in healthy subjects with a normal fasting glucose, it contributes like 60% on average to the glucose response after oral glucose. And with increasing fasting glucose concentrations, this is progressively lost. And again, it appears that this happens somewhere around the stage when we diagnose diabetes. Since we have traced the reason to the beta cell, let's see how the response to GIP compares in patients with type 2 diabetes and healthy controls. This is again in hyperglycemic clamp experiments performed with either no exogenous GIP, a small dose and a large dose, and the GIP concentrations reached are depicted down here. And you see much less response in the type 2 diabetic patients, and you can calculate an around 85% deficit here, which means you have only 15% of the normal response in patients with type 2 diabetes. And then I go back to very old experiments where the responsiveness to glucose is examined, and basically it is clamping at different glucose levels and then giving some other stimulus on top of that. And the higher the clamped glucose is, the higher the acute response is, even if you inject arginine at these different glucose levels, and much less happens with type 2 diabetic patients in both experiments. And here you have an 80% deficit. So that's quite close. And so our suggestion is what we see in terms of reduced responsiveness to GIP in patients with type 2 diabetes reflects the number and function of beta cells and probably is not specific to the GIP receptor. The story has been summarized in a review article, Perspectives in Diabetes, against Joris Meyer and myself as the authors, and we describe the typical findings regarding beta cells in type 2 diabetic patients, so like 60% reduction and amylin deposition. If you have less beta cells and you give a large stimulus, then they can no longer respond in the normal way, so this is flattening the response, which probably leads to some hyperglycemia. This may, according to some people, reduce GLP-1 secretion. We don't believe that, therefore 3 question marks. But it may reduce the expression of the GIP receptor in beta cells. And if you take all this together, then you can explain the reduced incretin effect in these subjects. Let me draw some conclusions. So in physiology regarding GLP-1, the gut produces GLP-1 in the same way and in the same quantity in patients with type 2 diabetes and in healthy subjects. This GLP-1 binds to receptors, for example, on beta cells, but there are other target cells for GLP-1 that I'll talk about in a minute. In physiology, this is very rapidly disturbed by the action of DPP-4, which removes the first 2 amino acids, and the resulting fragment, which is what we measure as total GLP-1, is no longer able to bind to this receptor and to stimulate insulin secretion. In addition, there is very rapid renal elimination of both the intact peptide and the fragment. And the half-life overall is just 1 to 2 minutes. GLP-1 has meanwhile been found to not only stimulate insulin secretion and reduce glucagon secretion, it has some other effects on the endocrine pancreas, like stimulating insulin biosynthesis, and in young rodents to stimulate beta cell proliferation and to inhibit apoptosis. It has motility effects, slowing gastric emptying. It reduces appetite and food intake. Has some specific activities on the heart, and of course with elevated insulin and lowered glucagon you have an altered glucose metabolism at the level of the liver, adipose tissue and muscle. And the miracle is that all these activities very well match the problems of our patients, like impaired insulin secretion, hyperglucagonemia, reduced beta cell mass, postprandial increases in glucose and triglycerides, obesity, cardiovascular disease, and high glucose levels, especially in the fasting state. So the miracle is that while GIP does not elicit any insulin secretory responses in patients with type 2 diabetes, although the activity of GLP-1 is reduced, it still has some effects. And you can make use of them therapeutically in these proof of principle experiments, taking hyperglycemic patients with type 2 diabetes in the morning, asking them to fast for another 4 hours, infusing GLP-1 or placebo. Placebo doesn't do very much, but GLP-1 will bring them into the normal fasting glucose range by stimulating insulin secretion, by suppressing glucagon, and with everyone responding. These are individual lines telling you that everyone in whom you start such an infusion of GLP-1 will have a lowered glucose. Those starting at very high levels of glycemia just need more than 4 hours to get to the normal range. There is some other special thing which applies to GLP-1, shown here, and GIP as well. If you look at the glucose dependence of this activity, even large doses of GLP-1 would not stimulate insulin secretion below fasting glucose levels. Only above a certain threshold, which may be 6 millimolar or so, you have a very steep augmentation of insulin secretion, whereas sulfonylureas, the other insulin secretagogues that we know much about, already stimulate insulin secretion significantly at hypoglycemia and normal fasting glucose, and they are not as well able to augment insulin secretion when the glucose levels are high. And this makes GLP-1 a very special insulinotropic agent because it cannot provoke hypoglycemia by itself. So 2 therapeutic avenues have been developed from these findings. The incretin mimetics, which are peptides like GLP-1. They look a little bit like GLP-1. They can bind to the same receptor. They should be resistant to DPP-4, and they should also have a long half-life. So now you know we have preparations that instead of having a half-life of 1 to 2 minutes, can be injected once a week. So this has been very successful. And we can inhibit DPP-4, which means for the same amount of GLP-1 secreted after meals, more remains in the active state and can stimulate insulin secretion and do all these other things that GLP-1 does as long as you keep DPP-4 activity low. So let me summarize. The question was, is diabetes a gut disease? And the answer is, diabetes mellitus is always accompanied by abnormalities in the incretin system. There is the reduced incretin effect, and we have the explanation. There is the reduced or even absent responsiveness to the insulinotropic activity of GIP. I hope I have convinced you that GLP-1 secretion is not generally impaired in type 2 diabetes, variable findings from different studies. We believe that the hyporesponsiveness of beta cells to GIP is acquired early in the course of diabetes manifestation. It may be related to hyperglycemia, but you all know that there are other abnormalities associated with diabetes, for example, free fatty acid metabolism. It could be cytokines, it could be inflammation of the beta cells or the islets. I think this is something we want to find out in the near future. We believe that GIP does not elicit an insulin response because beta cells don't function normally at this stage of diabetes. Quantitative considerations, and I've not gone into that very deeply, suggest that disturbances in the Entero-Insular Axis in type 2 diabetes contribute to diabetes progression, because if the cells neither respond to glucose nor to incretins, but incretins normally have a 2/3 contribution to the insulin secretory response. If you take that away, I think it's very logical that this will lead to the progression. For those reasons, GIP, in my view, probably is not a suitable parent compound for developing anti-diabetic medications. This has all been concentrated on GIP. And despite its somewhat reduced insulinotropic effectiveness in type 2 diabetes, it can lower and even normalize glucose concentrations. So it's not the etiology of diabetes that is connected with things in the gut, but the cure may come from the gut. And this is in form of incretin mimetics and of DPP-4 inhibitors. And since I showed you some original contributions, even unpublished work from my laboratory, I want to make you familiar with some names like Joris Meyer, Wolfgang Schmidt, the late Werner Kreuzfeld in Göttingen, and doctoral students, my dear colleagues in Copenhagen, Carolyn Deacon and Jens Holst. They always very exactly measure the incretin levels, and I have my group in Bad Lauterberg. And I want to thank all of them for their contributions to these findings. Thank you.